Hello, Internet. Welcome back to the Onco Data podcast or every Friday podcast. You may have noticed if you track with us, I've been a little in and out the past couple weeks hosting, but back to it and really happy to have another fantastic guest to join me today. Dr. Monika Lamba. I said Lambda naturally, and I messed that up. Dr. Monika Lamba Saini. Appreciate you so much. This is the first time we've met, and that's a great way to meet people is live on a podcast because it's 2024. So today we'll be talking about slightly different topics from our usual sort of trial focus, maybe clin tool centric discussions about data releases, and talking really more broadly in terms of pathology and applications of technologies in pathology, clinical applications of that, diagnostic applications of AI. So that's sort of the premise today. So super excited to have you, Monika. Again, nice to meet you. So if you wouldn't mind for our listening audience on the interwebs, could you sort of introduce yourself, your background, and then we could chat from there.
Thank you so much, Mark, for the invitation, to join you in the podcast. I am a pathologist by training and have worked in premier cancer hospitals in India, after which I decided to go to Belgium to do my Ph.D. in oncopathology, incorporating both molecular pathology as well as digital pathology. After my PhD, I joined Histogenics, which we presently know as Cell Carta, and was the principal investigator for quite some trials for oncology, for companion diagnostics in oncology. And I also owned my digital pathology and development of algorithm skills at Cell Carta, after which I moved to Scotland, Edinburgh, where I'm situated right now, with IQVIA laboratories, where I was the director of digital pathology at IQVIA laboratories. And at IQVIA laboratories, I helped in the deployment and validation and implementation of digital pathology systems across their laboratories, and also provided pathology input to develop AI algorithms de novo. After having spent almost 10 years in the clinical research organizations, I thought it would be a good idea to move to biopharma, especially now when we are talking of ADCs as the next big thing. So right now, I am the Global Translational Pathology leader for ADC Therapeutics in London. And I also take care of the scientific strategy and also look at translational biomarkers for them. And I also take care of let's say, using digital pathology to expedite the development timeline. So this was, in a nutshell, I think more than 20 years.
Well, I appreciate that. That's why I was sort of excited about today's podcast. Is you hit on so many things how do I phrase this, mentioned in our podcasts over time in such a completely different light. That's completely different, sort of what I'll hope is an initiation of a conversation to maybe revisit. I guess part of what I was saying is the word pathology is such an interesting one for me. Technically, once upon a time, my PhD work was in pathology, but that was very much preclinical cell. It was basically a cell biology perspective on very precise mechanisms of tumorigenesis. But then pathology can mean so many different things to so many different people. So it's one of the broader terms in science, I know. So,
and also, you know what, Mark, in the last few years, pathology, I believe, has really changed, and the way pathologists are working is also changing. Especially, I would say, we are in this Onco Data podcast, and we are going to talk about, I believe, AI and let's say some computational pathology, and looking at pathology in a very different way. Earlier, what used to happen was that, pathologists used to be like diagnostic oracles, and that would be the end of their involvement as far as the patient is concerned. right now, how things have progressed and how things have moved is that pathologists right now are a very important part of a team which helps to design the trial or design the treatment strategy or to design the biomarker. And I believe this particular development, especially when we talk of computational pathology and I mean diagnostic pathology, and incorporating computational pathology, I believe it is going to change the way medicine is going to be, uh, how we are going to perceive medicine in the future. So I think it is a very
Absolutely.
important time for pathologists to understand this shift, this change, and understand that this is basically once in a lifetime opportunity or once in a generation opportunity, I would like to call it that. That the way medicine is changing and is going to change in the next few years to come, I think it's once in a generation opportunity for pathologists to contribute to this change so that they have a seat at the table when this change is happening. And then all these algorithms which are being built up or all these classifiers which we are talking about shouldn't be a black box for them. They should be a part of the building of those algorithms, the building of those classifiers, so that they feel that they have contributed in this paradigm shift in medicine. That's my feeling about it.
Absolutely. Well, we're talking about a big universe, so I don't know where quite to begin. I mean, we talk a lot on our Onco Data podcast relative to our Clin tool, a lot in recent weeks about how we use AI in our systems, which are sort of, I think, fundamentally different than what we'll try to touch on super rapid fire today. We're really interested in looking at we with LARVOL extract public data and look at things like KM curves and look at meta-analysis and forest plots and what we can do with this type of data and applying AI to make outcomes predictions to making predictions about trial completion. But this this AI is a different universe. So I guess maybe if we could start sort of in the broadest sense. I know there's lots of, obviously you've been critical to the incredible just sheer technology of using image analysis tools on slides, which is sort of where my brain goes first as a once upon a time pathologist. But if we were to step back broadly, I'd say, what do you think are the most important applications with AI and maybe machine learning for image analysis and pathology? Hugely broad question, but sort of what's on the cutting edge is maybe a place to start.
To as far as I think that's a very good question, Mark. The thing is that image analysis tools are a very important part of digital pathology. Now, when we say digital pathology, it does not just mean viewing a slide digitally or viewing a slide on your laptop rather than seeing it under the microscope. It also means acquisition of that imaging data. It means management of the data, sharing of the data, and also interpretation of the data. So those things are very important. And when we are talking of sharing, interpretation, evaluation of that data, I think image analysis tools play a very, very important role. So basically, what are image analysis tools? Image analysis tools are computer-aided quantification of any histological characteristic of the tissue. Now, this could be in terms of morphology, or this could be in terms of, let's say, a biomarker identification or quantification. So when we look at image analysis tools in pathology, we basically characterize them into four, I would say, four broad categories. One would be quantification algorithms, wherein we quantify data, whichever in terms of, let's say, tumor volume or immune cells or percentage of a particular biomarker or a mitotic count. So these are just examples of a quantification algorithm or a quantification classifier. Then there are something called as detection classifiers, wherein we can detect an anomalous region or something like, a neoplastic tissue from a non-neoplastic tissue or a precancerous tissue from a cancerous tissue. So those things are also very important. They can be used as great screening tools. Then we have something called as characterization algorithms. Now, those characterization algorithms help the pathologist and then finally the physician also to grade the disease and to put it into a particular category, whether we are talking in terms of algorithms for grading a disease or staging a disease. So those are important as well. Then I think finally we have something called as predictive classifiers, wherein we know that how the patient is going to respond to a particular treatment or how the disease is going to shape up in the future with all other parameters put together. So these are basically, four, I would say, I would want to classify this into four broad categories.
So could you maybe help parse those last two for me a bit in terms of the algorithmic classifier versus the predictive classifier? So would you say, so is a predictive classifier more specific or broader? And in my brain, I think of specific markers looking at HER2 high versus low versus very low versus ultra low. I guess they're using new terminology for it. So when you talk about that predictive classification, is it on that level of specificity or is it the other way around? It's an incredibly dense question, but I wasn't quite sure what you meant by the difference of those.
Actually, if we look at it, you are right in a way when you are saying that when we look at the applications, let's go at the, very basic. When we look at the applications of these tools, we have something, as you said, that there are tools right now which have been approved by the regulatory industry, wherein we are looking at, let's say, quantification of a biomarker like ER, PR, HER2/neu, and PDL1, which is an immune biomarker, and many others as well. So these helps the pathologist to quantify it in a very reproducible manner. And some of them, like PDL1, are also used as a companion diagnostic, which helps in obviously deciding, whether the patient is eligible for a particular drug or a treatment. So this is basically, I'm talking of quantification classifiers. Then there are predictive classifiers, which will accumulate a lot of data together and then predict the response to a particular disease or to a particular treatment. So this could also mean that apart from the quantification classifiers, there is also cumulative data which can be added. So those really can help to look at how the patient is going to respond to a particular treatment. So those are also very important applications of looking at it. Also, I believe not only looking at biomarkers or not only quantifying these biomarkers, there are also analysis tools which help you to just look at the HE slide and then to quantify or extract information from it. Information in the sense that you have, let's say, how is the patient going to respond to a particular treatment, or what is the molecular profiling, or what could be the biomarker status of this patient. So these things are also being worked on just on the basis of HE, but obviously it can be challenging. There are also, I think right now, tools which are looking at virtual staining of slides to save tissue, but these are obviously in the pipeline. So, but there are people and companies working on it. And finally, I think one of your questions was, when we are looking at data all together, I think it's important right now, considering that when we look at diagnostic specialties like radiology, pathology, or let's say genomics, it's important that these diagnostic specialties are not just siloed. There is information and there is crosstalk between this data, which will help the clinician have a very, I would say, a very integrated report for targeted therapy, what we call as integrated diagnostics. So this is something for the future, but I think there are a lot of people who are looking at it with good interest.
So in the broadest strokes possible, in amongst all of these elements, where is AI showing its greatest success and where are its greatest limitations?
As far as the success of, let's say, machine learning or image analysis tools, I would say, is concerned, we do see a lot of acceptability in terms of quantification. Any morphological characteristic, or let's say a biomarker percentage, which can be quantified. Now, this could be this could be a biomarker in terms of percentages. This could be immune cells in terms of percentage. This could be a mitotic count, or this could be a tumor content. So, as far as that is concerned, I think the image analysis tools are doing a good job. Now, as far as the challenges are concerned, I think it's important that these algorithms or whatever classifiers we develop, there has to be more structure around the development of these algorithms so that they can be generalized from one laboratory to another. There should
Sure.
be more regulatory standards around digitization, around validation of the algorithm. And obviously, we need to have more collaborations and I would say crosstalk between a lot of, vendors or between a lot of players, where as far as image acquisition, image management, and also development of de novo algorithms is concerned. So these are things I think which there are still a lot of challenges happening. And I believe collaborations and seamless integration is important for all these things to work. So we need good IT infrastructure, we need personnel who are trained, and there should be more acceptability from even the pathologists to understand that this is just an adjunct or this is an assist to their work and not something which is going to replace the pathologist.
Well, I put you on the spot for such a big, broad concept, but I think that's a fantastic answer. It speaks to two terms that I think about on a daily basis. Is one of scale. I hate using the word scale, but for these, as you reference some of these algorithms and having them to be more broadly applicable takes communication technology, and sometimes I simplify that at using the term scale. And then that's what everyone's thinking about: AI and taking over jobs. So, not to
Yeah.
be silly about it, but I think everyone thinks about that regardless of
Yeah, I know
field. So,
that's quite a concern with the pathology community as well. But I believe the way those algorithms are designed, because I myself have been part of teams where we have worked at de novo algorithms. We understand that first of all, what works in one laboratory might not work the same way in another laboratory in different hands as far as staining is concerned, as far as annotations is concerned. So the generalizability of these algorithms is very, very important. Apart from that, the structural regulation around, all the software and the hardware, I think that also needs a good amount of regulation before any of these, I think, can be put into regular clinical use. So, but we do have
absolutely.
regulatory bodies right now looking at it. In fact, I think earlier this week, FDA came out with a regulation that, okay, all of these need to go through their own clinical trials to be just like we have clinical trials for other drugs or other biomarkers to make sure that these can be then used for clinical use, because there has to be a complete accountability and transparency for these algorithms.
Well, I think we'll have to take that new type of guidance for trials on these types of tests and algorithms for another conversation, because
Right.
I think we actually went over time a little bit. But as they say in social media and the podcast world, we're going to call that bonus content. So there you go. I hope everyone is having and will have a good Friday and a good weekend. Monika, thank you so much
Thank
for
you.
joining me to talk about a topic that's very much outside of our typical world of the software we build at LARVOL, but we're all in the same universe, and it was an absolute pleasure. And hopefully we can reconnect soon.
Right. Thank you so much again, and have a very good weekend.